right. So, I gave you guys back information on your experiment plans, which I already have the cells set up. They're going to be stimulated before lab tomorrow. So if you have any updates you want to give me to what I should be stimulating cells with or things like that, let me know when you can, because that's what I'm going to be doing this afternoon. The protocol for tomorrow's lab is already posted. There will be a one-page add-on that I will have for you tomorrow as well. Today I'm going to finish talking about B-cell selection, and then I'm going to move into talking about T-cells. If you look at the schedule, it says that we're doing an antigen presentation paper next week on Wednesday. I'm not going to be ready for it on Wednesday. Maybe on Friday. I will let you know kind of at the end of class this Friday whether I think the timing is going to work for that paper the following Friday. So we previously spent some time talking about aspects of B-cell development in the bone marrow. You can see some of that information summarized here. One of the things that you might have noticed that showed up on a lot of the past exams, if you were looking for past exams, was I often ask questions about this stuff with flow cytometry, and I didn't ask about that this time because I hadn't actually drawn it, the flow cytometry for this process this year, where I've drawn it in past years. So we can kind of take what we know about cytometry and apply it here. So we've talked about these three different types of cells that are found in the bone marrow: the pro-B cell, the pre-B cell, and the immature B cell. And one way that we could distinguish these cells is by using flow cytometry. And so we might imagine in this case, if I were going to actually look at bone marrow, I would find a mixture of all of these cells. We could draw what that would look like in a bit, but first I'm going to draw kind of what would happen if I had sorted each of these cell types. So I just had pro-B cells, or just had pre-B cells, or just had immature B cells. When we do this analysis, just like with any other sort of situation with a flow cytometry plot, we need to think about what are the two things we're measuring on each axis. On one axis, when we do this measurement of B cells undergoing B cell development, we'll look at heavy chain. And typically, when we say that, because it's the IgM mu heavy chain, we write mu heavy chain. We also could look at light chain. When we're thinking about light chain, there are two different light chains, kappa and lambda. I'm going to start this by imagining I have an antibody that binds to both light chains. Okay, so I'm going to say light chain. You wouldn't actually really do the experiment with an antibody that binds to both. You'd have to pick kappa or lambda. So we can think about how that changes the experiment in a moment. And so if I were to have a purified population of pro-B cells, and I were to measure them with flow cytometry, looking at the mu heavy chain and light chain, what would I expect that plot to look like? Yeah, Brenna. So Brenna says I would have negative, negative only. Why is that? Yeah. It has no BCR. Specifically, it has not yet made heavy chain, so it can't be positive for heavy chain. And it has not yet made light chain, so it can't be positive for light chain. So this is negative for heavy chain, negative for light chain. Now, what if we looked at a pre-B cell? Yeah, David.
Heavy chain positive, light chain negative.
So I would have a population of cells that are heavy chain positive and light chain negative. And why is that?
Pass on for pro 3.
Yeah, so the definition of a pre-B cell is that it has heavy chain on its surface. It's testing that heavy chain, but it has not yet made real light chain. And so it's going to look something like this. So what about an immature B cell? Yeah, Brenna? So now for the immature B cell, we're going to see this double positive population. An immature B cell has both heavy chain and light chain on its surface. And so this is what we would see. This would look, and so you could imagine if I did this whole thing and had actual bone marrow, had a mixture of these, I would see 3 groups of cells, and I'd be like, oh, those are the pro B cells, those are the pre B cells, those are the immature B cells, right? If I actually used an antibody that bound to one of the real light chain proteins, not just light chain in general, but let's imagine that I actually picked kappa, one of our 2 types of light chains. Would I see anything different with the pro B cells? No, because they haven't even made light chain yet, so why do we care? There's no light chain. How about pre-B cells? Would anything look different with the pre-B cells? No, because again, still haven't made light chain, so nothing should be looking different here. What about immature B cells? Am I going to see anything different when I look at the immature B cells? So I got lots of yes head shakes. What am I going to see different here, and why might I see that? Yeah, Jonah.
There's also going to be a space where we have a heavy chain without the kappa light chain.
Okay.
Because it could be
Yeah, so here, because we have 2 kinds of light chains, these are the immature B cells who used kappa, these are the immature B cells who use lambda. And so they could both be depicted here. And so I would actually get the same plot if I had total bone marrow, but when I saw this population, I would say this is either pre-B cells or immature B cells who used lambda. And I'd have to do additional work to sort of figure out which was which. Yes?
So just to clarify, for the immature, it could use lambda for the...
Yep. So with light chain, we have both kappa light chain and lambda light chain, and an immature B cell could use either. So you can see those immature B cells that have made both heavy and light chain here, still in the bone marrow. And the piece that we needed to go through in detail was this last part, or the central tolerance processes that will happen. Because as I mentioned to you before, when we do VDJ recombination, we're doing it in the absence of antigen, in this sort of sheltered area of the bone marrow. And we are making basically one of every combination out of all of the mini genes. So they can bind every antigen, even some pandemic virus that's going to hit you in 30 years. You already got a B cell that made that combo. And that's great because now you aren't going to have to wait quite as long to make a B cell, to evolve a B cell in response to that microbe. You already have one of everything. This is going to be really good evolutionarily to make sure that we have a very broad repertoire of B cells and antibodies. But the con of this is that when we make such a broad repertoire of B cells and antibodies, we also make some that can bind to self-antigens. And so remember that with antibodies and B cells, the antigen could be protein, carbohydrate, nucleic acid, or lipid, you have an awful lot of protein, carbohydrate, nucleic acid, and lipid in your body. And you basically, at the point of B cell development at this point, have made antibodies or made B cells with receptors that respond to all of those things. And so you could imagine if you let those B cells out of this nice protected area of the bone marrow to start trying to make responses, you would get destroyed pretty quickly. And have some pretty terrible autoimmune diseases. And so before we let those B cells out of the bone marrow, those B cells go through a testing process, which, as I told you guys before, is this process of central tolerance. So if they're not reacting with self-antigen, they get to leave, go to blood and other secondary lymphoid organs and do stuff. If they react with self-antigen, they're going to stay in the bone marrow and we're going to deal with them. Because and make sure that we don't have a self-reactive B cell that goes into the periphery. There are 3 things that can happen to the self-reactive B cell. And so I'm going to go through the 3 different things that can happen. I'm also going to tell you about some experiments that were done to kind of understand these 3. When they first started this sort of whole process and first actually did the experiment that I'm going to tell you about, they assumed that there was one option. And they assumed it was option 1. Spoiler alert, they discovered 2 and 3 in the experiment and found out it wasn't just one option. And so the first thing that can happen is what's known as deletion. So if we have that B cell that binds to a self-antigen in the bone marrow, that B cell, and this is particularly true if that B cell gets a really, really, really strong signal. That B cell will be signaled to undergo apoptosis and will be deleted. We're going to kill that B cell, not let it out of the bone marrow, not let it go any further because it is just too risky. And this was, like I said, sort of the idea that people had, and that this is what happened for a long time. But some people would argue with this theory a little bit. And they would say, well, okay, so we're going to look in Dr. Barker. We're going to say she's been making B cells all her life. And we're going to see if she's ever made a B cell that responds to actin. Okay, so we're going to look in her body and see if we can find the B cell. And if we can find it, that means it's made. And if we can't find it, it must have been deleted. What's the problem with that logic? There is a big problem with that logic. Yeah. So if it got deleted, how would you see that it was made? And that's basically what we're saying, but there's another way of thinking about that. How do you know that you just didn't find it that day? Are you actually going to have a test tube with every B cell from my entire body? And are you actually going to look at every single one of them? This is kind of a needle in a haystack problem. How do you say, I didn't find this rare thing, so it doesn't exist, versus I didn't find this rare thing because it's rare and it's hard to find? And so people had to come up with a way to kind of change the numbers a little bit so that they could definitely know the cells were findable and definitely track the cells and really see there should have been a lot of these cells and now there's not, they're missing or something like that. And so this is the experiment that they ended up doing. And in this experiment, they are using some mouse strains. I went back and forth about whether I wanted to go through this experiment or not. And I think one of the big reasons I decided to do so is because I wanted to introduce mouse strains, because with some of our next topics, we're going to see so many mouse strains that would be good to talk about. So generally, immunologists do a lot of work with mice. And one of the big reasons why we work with mice is that we use mouse strains, which are genetically identical mice. And so if I have multiple mice, say of a strain called BelbC, their entire genome is known, and every BelbC mouse has identical genomes. They actually were inbred, for many generations since like 1900. And so they all have kind of identical genomes. And so that's one reason why we like to use mice. But the other reason why we like to use mice is because we are really good at genetically modifying those mice. And so we can do all sorts of genetic modifications to our mice to help us do tricky experiments. And in this experiment, the authors did a couple of different modifications to their mice. So, I'm going to start with this mouse that's over here on the right that they made. So in this mouse, they inserted 2 genes. One of those 2 genes was a rearranged heavy chain. So VDJ, all rearranged, all set, for an antibody that they were interested in. And that antibody bound to an antigen called HEL, which I'll mention a little bit more about in a second. A heavy, and the second one is a rearranged light. So basically they gave the mouse pre-rearranged heavy chain, pre-rearranged light chain. And when you do this, you actually do this in before the mouse is basically an embryo. So every cell of the body has this genetic change that you've made. So that means all of the skin cells and whatever of the mice, and we don't really care about them. But we do care about all of the B cells that are developing in this mouse. So when those B cells are developing, they already have a pre-rearranged heavy chain gene. They don't have to do rearrangement. And that pre-rearranged heavy chain is going to get transcribed and translated, and it's going to go on the surface of the cell. And so they're going to skip right on through that pro-B cell stage and be pre-B cells. And they're going to be like, do I have a receptor that works? Why, yes, I do. I didn't even have to do any work to get it. And that they're going to, in fact, allelically exclude so that none of the rest of the heavy chains can ever rearrange in any B cell in that whole mouse. Every B cell in that mouse is going to be like, hey, look, I won the lottery. I found a heavy chain and didn't have to do any work for it. And then same thing, when that cell tries to become a immature B cell and rearrange the light chain, it's already going to have a pre-rearranged light chain. And so in the end, in this mouse, this transgenic mouse with the anti-HEL antibody genes, 100% of the B cells that develop are going to respond to this one antigen, HEL. This would be somewhat terrible if this mouse escaped the lab and ran through the streets of New York City. Because this mouse would have a B-cell compartment and antibodies that could only protect it from this one antigen, HEL. And so if it got infected with any pathogen, it would die. So this is not something you find in nature, but it's something that we do a lot in the lab to help us find a lot of a particular cell. Because now, if we want to actually see what happens to the anti-cell HEL B cells in this mouse, we're not looking for one cell. We're looking for all the B cells in the mouse. So we don't have to worry about can we find it or not. HEL stands for hen egg lysozyme. And the only really important part for you to know about hen egg lysozyme is that it is a protein encoded by a gene from an animal called a hen, aka a chicken. So if you think about this mouse, does this mouse normally make this chicken protein, hell? No. So that's a foreign antigen to this mouse, right? Basically, 100% of this mouse's antibodies make antibodies against this chicken protein. If it got attacked by a chicken, it'd be in great shape. So the mouse lives happily ever after. They also made a different strain of mice where they added a gene to the mouse's genome that encoded this chicken protein. So we also have this mouse over here that is making the chicken protein now. This mouse probably had one B cell undergoing B cell development that responded to the chicken protein. And we're probably never going to find it because it's really hard to find just one B cell. So it's going to be really hard to track. And the mouse probably doesn't care because it also has the rest of its B cells that are going to respond to other stuff. And then these authors took their mice and they crossed them. So they made progeny mice that started to make the hen protein, the chicken protein, and that had 100% of their antibodies that would respond to the chicken protein. So now, in this progeny mouse, 100% of the antibodies in the mouse bind to an antigen that's also in the mouse. So now, before it was 100% of the antibodies bound to a foreign antigen, but now 100% of those antibodies are binding to a self-antigen because you've made it a self-antigen by adding this to the mouse's genome. So now we can track, well, what happens to those B cells? And so they did some flow cytometry experiments. So first, they looked at the first plot that you see on the left, which is B cells that bind HEL on one axis and just IgM, aka B cells, on the other axis. And first, they just looked at a mouse from the mouse store. And you can see they basically had two populations of cells that they saw in this experiment. They don't show it as dots here, so you might not be as used to looking at the contours. They've got one population of cells that they see right here. I would put those as a population there. Those cells are not B cells, and also not B cells that bind HEL. So those might be T cells or some other not B cell that is in these mice. They also had some B cells, and most of these B cells did not bind HEL. So these are just the B cells that don't bind to that antigen. This experiment was done in the 80s, I believe. If we did the experiment now, we would also see, look, one dot up here for the one B cell that does bind HEL. But they weren't good enough to find the one individual cell here. So we might have the one B cell up here that bound HEL, but it would be really rare. And this is, again, just in the plain mouse before we made any modifications. Yeah, Jonah.
I was going to ask, when you have the mice that produces the HEL
Mm-hmm.
protein, wouldn't a B cell that would, because isn't that all B cells can find one extracellular thing and it would start to make copies of itself?
Maybe. And so, but the other question is especially because when they are in this experiment, they're assuming deletion. So what they're kind of assuming is that one B cell was deleted and didn't get let out of the bone marrow. And they're doing this experiment looking at the blood. And so they made the one B cell, but then the one B cell got deleted. And so they're never going to see it.
Okay.
Is kind of what the assumption is here. I'm going to come back to that in a second, okay? So this is just a plain old mouse. This is just average mouse. They also looked in your plot that's in the center of this, same axes. At B cells that, they're looking now in the mouse where they added the heavy and light chain for the B cells. And so now in these mice, they have some non-B cells. And all of the B cells bind to this antigen HEL. We don't have any B cells who don't bind to that antigen. Okay? So, if they looked at the mouse, so this is the one with the HEL antibody genes. This is the one just that has HEL antigen. IgM B cells, B cells that bind HEL. Jonah, what do they expect to find in this mouse? Is basically what you just asked me. So what are they sort of assuming that they're going to see in this mouse? No IG. So we got these. So we got some not B cells. Okay. What else? Yeah, Brenna. So this is the one where they just have the antigen. So only they should have, in theory, maybe only ever made one of this B cell. So they should have a whole bunch of B cells that respond to other stuff. And maybe they have the one B cell, or maybe they don't because it got deleted. Either way, this is in the 80s and you can't see that rare of a thing on your flow cytometry plots. So they're not going to look different at all. Okay? So then here's their question. Now they have the progeny mouse. The mouse that is the progeny of this one and this one. Now it has 100% of its B cells are going to recognize this one antigen, HEL, and that one antigen is a self-antigen. Now, what do they expect? And again, before this paper, they thought the answer was deletion, and deletion was the only choice and there were no other choices. So if deletion is what they are going to see, what should they expect in their plot? Yeah, Brian. They're going to have non-B cells still, but they're going to have no B cells because all the B cells should recognize HEL and they should have been deleted. So this is what they were expecting to see in their experiment. Before I go to the next slide, which is actually their data of what they saw, I have one other thing that I have to tell you about their experiment. When they did this experiment, they actually did the HEL antigen mouse, the mouse that makes the antigen, in two different ways. So one way that they made it is where the HEL antigen was on the surface of cells. It was a transmembrane protein. And one way they made it was that it was a secreted protein. So it sort of was outside of the cell. We now know that these two types of antigens differ in terms of the strength of signal they give to a B cell. This one might bind to, say, multiple B cell receptors or multiple times and might actually give a really strong signal to the B cell because the two antigen copies are right next to each other on the surface of the cell. This one is probably going to give a weaker signal to the B cell because the secreted proteins are like off floating around. So actually they're probably not all like next to each other, like 4 triangles there. Really there's like 1 triangle here and 1 triangle here and 4 triangles around. So there's not one B cell that's going to be able to reach all those. Yeah, so the idea is that the triangles are relatively close together. They're all held on the surface of the same cell. And so one B cell is probably going to get stimulated by a bunch of them at once. And so it's going to be a strong signal. The one where they made it a secreted protein, they're honestly not going to be next to each other at all. So that you're probably not going to get a very strong signal. So they actually set this up in 2 different ways. We found that the answer of what might happen to the B cell changes a bit based on signal strength. And so these are the data that they got. And all right, so we're basically looking at what we've been drawing on the board, those kind of plots at the top. At the bottom, we're actually using 2 different markers for B cells. And so, you really should hope that you have, are there B cells or not on here? And so it's B cell versus B cell. Hopefully you don't have cells that are B cells but not B cells or something. So here's the mouse that was the regular old mouse from the mouse store. It looks like exactly what we drew over there. 2 populations. Here's the mouse with the HEL antibody genes. Looks just like what we drew, 2 populations. Here is the mouse that is the transgenic that we made. And in fact, this is the membrane version of the antigen. So that was our hypothesis that we drew on the board. What do they see in the experiment? Yeah, Jonah. So we have 2 things that we can observe here. One, there's B cells. And you can also see that down here. There's cells that are B cell positive. So your hypothesis was that all the B cells, there were going to be no B cells because they all got deleted. So one thing we can see is there's B cells. They didn't all get deleted. And the other thing we see is that those B cells don't bind to our antigen. Hell, even though we gave them the genes and they're supposed to 100% bind our antigen, they're B cells that don't bind the antigen. And this told them, okay, so if we have a self-reactive B cell developing in the bone marrow, it can change what it binds. It can change the thing that it reacts with. It's allowed to swap what it reacts with. And this is literally how they figured that out, is that they did an experiment like this and they're like, what in the world? The B cells still exist and they bind to the wrong thing?
So it looked no different than the antigen diagram? When they added genes, not the chains?
Yeah, I mean, it looks basically like the same as the normal mouse also. Yeah, Josh.
What about the measles that were already made before the gene was put into the...
So you actually are doing this on a embryo. So there are no B cells made before. This is all done in the embryo, and then you develop out that embryo. So every B cell is made with this chain already rearranged. Yeah.
You're only changing the heavy chain.
No, heavy and light. Heavy and light. Yeah. Yeah.
So it shows that B cells that are separately cannot leave the bone marrow? Or that they
No, so it says they can change and then leave the bone marrow. So I told you one option was deletion. There's the second option, which is changing before you leave the bone marrow. This option is called receptor editing. And so what we've seen is that sometimes, that B cell can just die, but sometimes that B cell can actually try to make a new receptor, test again, and be like, all right, now you're good, and leave the bone marrow. And this is a process known as receptor editing. One of the things that is really key to note about receptor editing is receptor editing only happens with the light chain. So we can't change our heavy chain, but we can change our light chain. And so what you can notice here is, here's my B cell. It has black heavy chain, black light chain. It's binding to a self-antigen. And so now it might swap out and make a new light chain, like blue light chain or pink light chain, and no longer be self-reactive. Because remember, your reactivity is about the combo of the heavy and the light chain. Now you're not self-reactive, you're allowed to leave. And you can see this receptor editing process happening here and realize kind of what's going on with the light chain. So let's imagine our B cell picked V3 and J3. You can see we have this V3, J3 join in the light chain. And let's say that B cell gets a signal that says, you are self-reactive. Bad. That B cell can actually do another light chain rearrangement if it has any outside V's and outside J's left, and delete that intervening self-reactive chain and use a different pairing now of light chain genes. And so you can see this happening here. Here's our self-reactive B cell. It used this V and this very lightly colored J. I don't know why all the V's are the same color. That's not helpful. It was self-reactive. It's sad. And so it might now rearrange again with an outer V and an outer J, delete the one that it had before, and make a new light chain to go forward. This is another situation where the cell will have RAG on. And B cells can actually receptor edit quite a few times, depending on how many, like if they happen to use, this B cell happened to use V3 and J1 the first time, if it happens, and again, this is all kind of a math probability thing, if it happened that it tried again with V2 and J2, and that one was self-reactive too, it could try again. It can keep trying, and so B cells get a lot of tries to make a light chain that doesn't give them self-reactivity. And in fact, they get to try on both kappas and both lambdas multiple times. So they get a lot of chances to make a good light chain that's not self-reactive. And then be allowed to leave the bone marrow. So there's one other part of this to think about, or one other piece of this to think about, which is that if you notice, I've mentioned this multiple times, we are only going to receptor edit at the light chain. We are not going to receptor edit at the heavy chain. We do not have a chance to make a new heavy chain. And so if like the reason why you're self-reactive is really because of what heavy chain you picked, you can try the light chains you want. And it may be really hard. And so we need to think for just a second about why we cannot receptor edit at the heavy chain. And so here's my light chain rearrangement at the top. Here's my heavy chain rearrangement at the bottom. And I am going to ask you, why can we not receptor edit at the heavy chain? Yeah, Shelby. Yeah, because when you actually make a heavy chain, you end up deleting all of the other Ds. So you might have some Vs and Js that are remaining, but there's no D. You've cut out all the Ds, and the V and the J don't have RSSs. Like they have the same kind of RSS, so you can't do the 12-23 rule and put those together. So you're out of luck. You're stuck with the heavy chain you got. Yeah.
So we were talking about like the two options before in the die or change.
Mm-hmm.
Does it only die if it cannot change?
We think so. To be perfectly honest with you, the tracking the cell that dies versus changes is still actually kind of a hard problem. And so we know some die, but there's certain details that we're not totally sure about. But I would say yes, it can try multiple times. I would think it's probably based on like signal strength again. But exactly why the cell chooses die versus receptor edit, I don't have an answer for. But they did one other experiment as well. And so here I'm showing you the exact same data that I showed you previously. So this is their experimental data again. So same thing. We got the regular mouse, non-B cells, B cells. The transgenic mouse, non-B cells, B cells. All the B cells bind hell. The double transgenic mice got non-B cells. And we strangely have B cells that do not bind hell, which we were not expecting and is a fabulously interesting experiment. But they also repeated this experiment with the antigen made in a different way, the soluble hell. And so what you can imagine with the soluble hell is giving a weaker signal. So the signal is not quite as strong of a self-antigen. And so they also did that experiment. And these are the data that they saw. Again, exact same plots that we've been looking at. So what did they see? What did they observe in these mice that had the soluble antigen or the antigen with weaker binding? What did they observe? Yeah, Brian. Okay, well, so we're gonna get to that. Well, so, but what do they observe? Like, what cells are present? What cells are absent? What's going on with them? So they got B cells, and the B cells still bind to this antigen, even though it's a self-antigen. And we're doing this experiment in the periphery. And so we said, oh my gosh, if it's a weaker binding to that self-antigen, you let the B cell out of the bone marrow. That's weird. That is definitely not what we were expecting, is that we're letting these self-reactive, as Brian sort of hypothesizes, B cells out of the bone marrow. We're letting these B cells out of the bone marrow. What's the next question that you might have about these mice? Yeah, David. Were they healthy? Did these mice have like rampant autoimmune disease? You know, because they have all these self-reactive B cells now in their periphery, right? The answer is no, they're perfectly healthy, no autoimmune disease at all. So somehow we're getting around these mice having autoimmunity, even though they have these self-reactive B cells in their body. And this is how they discovered the third option that can happen. And this third option is known as anergy. The basic idea, and so here you can see if we have high binding, we either get deletion or receptor editing. If we have low binding strength, we can get anergy, or we get an anergic B cell. And what's really important to remember if we think about these data, and everybody actually gets this when you think about, well, there are parts of it you get and parts of it that you hate in general when I talk about this. What this graph shows you, what these data show, is that those B cells were present in the mouse. What it does not actually show you is whether those B cells had a function and whether those B cells could do anything. If you think about all of the experiences in all of your life, I will not say about, when you're in different classes or whatever, you can think about whatever experience you want. You might imagine a situation where you have been present, but not functioning. Present, but not functional, right? And so this tells them those cells were present. But when they did further experiments, they actually found out these cells couldn't work. These cells didn't function. And so often when I think about anergy, I think about the idea that the cell is basically completely turned off. So we'll let you leave the bone marrow, but we're going to turn you off and say you can't do anything and make you useless before you're allowed to leave the bone marrow. And so here you can see this as well. We have our B cell that's getting sort of this weaker signal. The B cell is going to become unresponsive to antigen. It's going to enter the peripheral circulation and it is going to be unresponsive, completely off, completely unable to respond. So it's going to be an anergized cell. So the cells, again, are present. They're able to leave the bone marrow, but they are turned off for safety purposes before they leave the bone marrow. Yes. I'm guessing, there's a question that always comes up here, so I'm sort of guessing that's what it is. Yes.
What's the difference that, how do they know that it still binds to the antigen or really because it's not binding?
Well, so they can do experiments like this, and they say, oh my gosh, the B cell's present, and it binds antigen. And so they can see B cell's present and binds antigen. But then if they do further experiments, like in the lab, and say, can I make it function? They can't. And so I'm not showing you those data. Does anyone have any other questions about anergy? Sorry, like I said, there's a sort of a question that comes up here, so.
What purpose does it serve?
Why? What's the point?
But it's just like floating around.
It's just the what? Why? Yes, that would be the question that usually comes up here. So I want first for you to think about sort of B cell development with these three options. Of these self-reactive B cells that are drawn up here, that are sort of going through this process, which one's the most dangerous? The one that binds really tightly, right? So we're in a situation of the kind of slightly less dangerous cell, not the most dangerous situation. So let's imagine that you were involved in security for something. Like you work for the TSA. You're gonna have to pick a line of this is okay, this is not okay, right? And if you make your line really, really, really tight, then you might actually prohibit some things that shouldn't be prohibited. And if you make it really, really, really loose, you're probably going to let through some dangerous stuff, right? And so here, it's kind of about, okay, so we're going to definitely not let these super dangerous ones out. These ones that are a little less dangerous, we're going to actually let out. And there's a reason why we're going to let them out. Let's imagine that you, the organism who houses these B cells, has a ridiculously terrible microbial infection and you're going to die. In that situation, you want every B cell possible who could participate. Even the ones that are kind of a little bit dangerous. If they could help, you need their help. In the super total crazy emergency situation, you want to have everybody that you could possibly have around. Even the ones that were maybe a little dangerous. And so I tell you here that these cells are unresponsive in the periphery. In reality, I want you to think of them as unresponsive cells. I want you to imagine them. If I ask you about anergic B cells or later when we see anergic T cells, but whenever you see anergic something, think about unreactive, completely off, never going to do anything. In reality, if they get an absolute sledgehammer of a signal, like so much cytokine, so much, they can turn back on. Because it's in like the total emergency last ditch situation, they'll turn back on maybe to help. And so the idea is that we're letting them out, we're keeping them off in all of normal life. But if there is some insane amount of cytokine situation where you are in such an emergency, they could potentially turn back on and participate in the response. In some of the cases where people seem to have long COVID, they actually are seeming or have autoimmune diseases after certain infections. It is actually because we think some of their anergic B cells or anergic T cells got turned back on that shouldn't have. Because like during their COVID infection, they had so much cytokine that all the alarm bells were ringing and everybody got turned on. And now there was autoimmunity where there wasn't before. And so, what I want you to know about anergy is think of those anergic cells as cells that are released into the periphery and turned off. The question always becomes, well, what the heck is the point of that? And the idea is that you can imagine them as being like total last ditch effort reserves that we're keeping around. Yeah.
So they only get turned on if they're like out.
Yeah, they get turned on out of the bone marrow. They get turned on only if in this crazy emergency situation. I would imagine that does not happen. And when you're imagining like the basics of what this looks like, that should not ever happen. Yeah. Everybody's going to get energized coming out of the bone marrow. And then it's just if for that 2 days, if you get so much cytokine, then, so the new ones will probably come out energized. Yeah. So this is where we have to think a little bit about some basic biochemistry. They might not be perfect binders, or they, especially, let's imagine this. I'll just draw a shape. I don't need a cell. Okay, there's my self-antigen. And let's imagine I have an antibody that has binding site that looks like this. Okay? It could bind to this kind of weakly. It's not perfect, but it's okay, right? It's a weak binder. Now let's imagine that there's a pathogen that has an antigen that looks like this. Even though it was a weak binder to self, it's actually going to be a great binder to pathogen because there's only 20 amino acids. There's only so many ways you can pair them together. So self-antigens and pathogen antigens might not actually be all that distinct. And so maybe you're in this emergency situation where this pathogen is around. And you're like, all right, I got to even turn on the risky ones. And now you turn on this B cell that makes this antigen, and it actually is the one you needed all along. Does that make sense? It's uncomfortable because it's another situation where you're like, wow, the immune system is really inefficient and wasteful and does a lot of wacky things. But if you really think about how you would evolve a way to have as broad recognition as possible, you end up with things that look like this. When we think about central tolerance, whether I'm thinking about central tolerance of B cells, like we're thinking about here, or whether I think about central tolerance of T cells, which we'll talk about later in the semester. One thing that comes up, and this in some ways is a little obvious, because I told you there's this thing called peripheral tolerance, where we have tolerance stuff that happens later, post primary lymphoid organ. In both the B cell case and the T cell case, there is a little problem that comes up with central tolerance. They're slightly different problems. And when you hear these problems, this is especially true with the T cell problem, you say, oh my gosh, that's really bad. How is it that we're alive because of that problem? Because you look at the problem and you'll say, oh my goodness, Dr. Barker, I think that there are a lot of B cells that could slip through that crack that you're going to tell us about. Or again, when we get to the T cell one, basically everyone spends some time being like, I'm sorry, how are we alive? If that's a crack in this central tolerance mechanism. And the real answer is, well, that's why we have peripheral tolerance too, to sort of act as another layer to catch this stuff. But I want to point out for you where the crack is in B cell central tolerance. And again, there's reasons why I point this out and reasons why it's really important. So let's see. This will work. Here's our B cell. Our B cell is getting tested for against self-antigens. We're doing our central tolerance testing, and then we're going to let the B cell leave the bone marrow. And let's imagine that we have a B cell, and it has a B cell receptor. They probably do. We're imagining one right now, though. Okay. We're imagining a B cell. It does all of its VDJ recombination, and it binds to, I don't know the name of the protein, but we're just going to make it up. It binds to the protein that makes my eyes blue. It binds to the protein in the rods and the cones that lets me see. So imagine some rod cone protein that lets me see that's in my eye. Did that B cell encounter that rod cone protein in the bone marrow? No, because it's only made in the eye. And we're looking at the bone marrow. And so we are going to have great central tolerance in B cells for antigens that are found everywhere in the body, like, actin or something like that, or things that are found in bone marrow. But if we're talking about an antigen that's only found in one particular location in the body otherwise, the B cells don't get a chance to test against it in the bone marrow.
Is that because they haven't reached that antigen?
Yeah, they haven't reached that antigen. They don't get to test on it. And so you can think about it and you can be like, oh my gosh, there must be so many B cells that slip through this crack. Oh no. How do I not have autoimmunity? And the answer is peripheral tolerance. But the answer is also, that's partially why the frequency of autoimmunity in people is higher as they get older, is because there's more chances for this crack to be slipped through. I also sort of have described all of this to you as the bone marrow is this nice separate place, the sort of testing place. I have assumed that there are no pathogens in the bone marrow. And in fact, there are very few that end up in the bone marrow. So you can really imagine there being no pathogens in the bone marrow. But what would happen if a pathogen got into the bone marrow? Yeah, Shelby. Yeah, the B cells would be tested against that pathogen, and what would happen? They wouldn't react to it. They'd be like, oh, this is a self-antigen. Can't respond to this thing. Better let this thing alone. And so, in fact, if we ever had an infection and had a pathogen in the bone marrow, that would be pretty bad because you would induce central tolerance to that pathogen. And fortunately, we don't really have a lot of pathogens that end up in the bone marrow. So everything we've talked about thus far with our B cell has been something that has happened in bone marrow. And only when they have gone through all of these processes will they go into peripheral lymphoid tissue and start looking for antigen. And so to clarify something that might confuse people sometimes, the B cells here, the B cells in the bone marrow, you can think of them, they're like the B cells at school, learning stuff. They don't kill stuff. They don't attack stuff. They're not making antibodies. This is just like school time. You don't actually do anything about an antigen until you're out in the real world of the periphery. We have basically followed our B cell from stem cell to leaving and going into the peripheral lymphoid tissue. One thing I want you to know is that when that B cell leaves the peripheral lymphoid tissue, turns off RAG is never coming on again ever for the rest of the B cell's life. We are done with RAG. And now our B cell has sort of transitioned from the yellow area and the yellow functions, doing diversity stuff, getting checked, to now being in the pink areas where it can do the pink things, like finding microbes, attacking microbes. And so now we're kind of switching which part of our B cell's life we're in. For the majority of these functions, especially the parts of these B cell functions that I think about a lot, like being a memory B cell, or making a lot of antibody as a plasma cell, or all sorts of doing class switch, doing affinity maturation, all sorts of, those fun B cell functions. They all require the B cell to be activated in the periphery. And one of the parts of B cells being activated in the periphery is that they have to interact with a T cell. So here's a T cell interacting with a B cell. So in reality, for our B cell to do anything once it's left the bone marrow, yes, I know B cell biologists who may be listening on YouTube. I am skipping some stuff, but that's okay. In general, that B cell really needs to interact with a T cell. So I actually can't tell you anything else about the steps of the B cell's life until I tell you about T cells. Because for the rest of the B cell's life events to happen, they need a T cell too. And so this is why at this point we switch gears into thinking about T cells. Basically, I am a full thing behind because I'm getting ready to start the T cell. The next slide is the T cell slide, and I have or starting T cell stuff, and I have 1 minute for that. So I'm going to basically tell you that starting next time, we're going to start talking about T cells. And in doing that, we're going to first think about some of the immune responses we've already seen and why they're not enough. Why this is not enough immune system. We've already had a lot of immune system this semester. Why do we need more? What situations are still not covered? And we're going to start talking about T cells and this associated protein called MHC. And most of what we're going to say was about MHC. I will also tell you that the next lecture involves a lot of fun facts about MHC and might be my favorite lecture overall because the fun facts are so fun. So be ready.